Good morning, everybody. So we'll continue chapter number second, that is sexual reproduction in flowering plants. So as we discussed in our previous video, in that PPT presentation, the two important methods of the sexual reproduction in flowering plants, androecium and gynecium. Okay, that all the details we have been discussed, how the embryo is formed, what is TMC, what is MMC, so all the things we have discussed. Now we are going for the further topic, the next topic, that is pre-fertilization, and in that is pollination. So structure and function we are going to discuss. So each video will be bigger one for you because we are discussing each and every corner, each and every points for this chapter, for this topic. Okay, so here pollination, my dear students, we are aware what is pollination. It is just transfer of pollen grains from the anther to the stigma of pistil. So pollination is a process, is a transfer of pollen grains from the anther to the stigma of pistil. So that we are aware, and we have been discussed. We already know that the two type of pollination, self pollination and cross pollination. So that also we will try to discuss. So here the pollination on the basis of the types. So here if you see, we have generally three types of pollination. Recently we were aware the two types of pollination, self pollination and cross pollination. Now we have the scientific method in that on the basis of the source of pollen, the pollen condition is three types. So here autogamy means self pollination, and here this is geitonogamy and xenogamy. So two again autogamy that we call as self pollination. These two are cross pollination. So geitonogamy and xenogamy. So these three you need to remember. So three types of pollinations: autogamy, geitonogamy, and last is xenogamy. So here we will discuss one by one each and every type of pollination. First we will take autogamy. Autogamy means itself. The word itself indicates auto means itself. So the process will be carried out from anther. The pollen grain will be transferred to the same flower, to the same species on the stigma. So that we call as a self pollination or autogamy. And here it is a transfer of pollen grains from anther to the stigma on the same flower. Okay, that we are not going in detail, but general concept we must be clear. So that is the same flower, the anther will be transferred to the stigma, and the pollens will fall here, and the fertilization process will start. So that we call it pre-fertilization. And here autogamy, different examples we are going to discuss. The plants like viola, it is common fancy, zeolis and camelina produce two types of flowers. In that plasmogamous flowers and clistogamous flowers. Two types of flowers they produce in the autogamy: plasmogamous flowers and clistogamous flowers. So here you can see clistogamous and plasmogamous flowers. The clistogamous flowers may be at the root part, okay, maybe root part, and this plasmogamous flower at the top. It may be available in the stem region. It will available in the branches region. Okay. So here plasmogamous flowers. How we can easily identify? How we can easily understand that this flower is of plasmogamous flowers or clistogamous flowers? So here types are similar to the flowers of another species with the exposed anthers and stigma. They are similar to flowers of other species with exposed anthers and stigma. So here easily we can understand by looking at the flower, anthers and stigma, we will easily identify the flower is of plasmogamous. Okay. Next is the autogamy. In the next flower, that is clistogamous. They do not open at all. So in that flower is not getting open. We will easily identify. If flower is open, we will name that is a plasmogamous. If it is not open, there is a clastogamous. Plasmogamous and clastogamous, that we will easily understand. And they are autogamous. Clastogamous flowers are autogamous, and anthers and stigma lie close to each other. They are very close. They are very near. Okay, and plastogamy leads to inbreed depression. The process occurs inside because it is totally closed. So that is why it is inbreed depression. Geitonogamy. It is the transfer of the pollen grains from the anther to the stigma of another flower of the same plant. Understood? It is generally cross pollination. Geitonogamy. The same flower, same plant should be there. So it is a transfer of pollen grains from the anther to the stigma of another flower of the same plant. Here, generally we take example this jasmine we can take. Okay, so it is a functional cross pollination involving the pollinating agents, but it is genetically similar to autogamy since the pollen grains come from the same plant. This is the process which carried out on the same plant, same flower, but it will be cross. Got it? So like that, it is a kinetogamy. Next is a xenogamy. The xenogamy, it's a transfer of pollen grains from another anther to the stigma of a different plant. Different plant I am talking about. It brings genetically different pollen grains to the stigma. So plants are different. Here another you can see that it will be transferred to the different plant. But that two processes occur in the same plant, but only the xenogamy it's occur in a different plant. Now the agents of pollination. I think I have to remind you, in 4th standard the agents of pollination were there. The germination, name of the chapter, the plant in that process was germination. Okay, so in the dispersal of plants, the agents we remember, if you know, in that two agents were there, biotic agents and abiotic agents. So biotic agents in the insects are there, mostly live things are there, insects, birds, bats, primates, arboreal, rodents, reptiles, or many more, even humans also. And abiotic agents being water, then different air. Okay, so that explosion, so different type of abiotic agents were there. So here we will discuss the agents of pollination by one quickly. So here abiotic agents, first we will take wind. Here we can take example corn tassels. Okay, makai, we call it makai. So in that it will be transferred with the help of wind. So pollination by wind is called anemophily. Anemophily. Understood this word? The pollination by wind is called anemophily. Pollination by wind is called anemophily. And more common abiotic agent, that is, we'll take example, and they having a fluorescence, example, corn cob. The tassels are the stigma and style which wave in the wind to trap pollen grains. You can see. Generally we are playing also. Sometimes we are playing with this also. We are doing like this, you know. So wind pollination is quite common in grasses. Generally, most of the grass, this type of corn cob will be tassel from one place to another place. Okay, so that the pollination by wind. Second, the ways for the effective pollination. So flowers produce a large amount of pollen. Pollen grains are light and non-sticky. They often possess well-exposed stamens for easy dispersal of pollen grains due to the wind currents. The large feathery stigma to the trap airborne pollen grains. So here you can see. Okay, the structure you can see, you will easily understand because they are very light. That is, then only they can able to move from one place to another place. They can fly. Okay, so that is why the wind. Next, water. Water is very common. Vallisneria, Hydrilla, and Zostera. Vallisneria, Hydra, and Zostera. So this type of plants are easily transferred, easily float, if you know that, on the water. And from one, just example, you can take coconut. Okay, coconut you can take. Generally, we were using this word, this example for the pollination by water. That example we were taking, that is coconut. So that also you can remember. Just different detailed study we are going to remember. Here you can see it is quite rare. It is limited to about 30 genera, mostly monocotyledons, and the pollination by water is called hydrophilic. Okay, hydrophilic. Pollination by wind is called anemophilic. Pollination by water is called hydrophilic. Remember? So here Vallisneria, the female flower reaches the surface of the water by the long stalk, and the male flowers or the pollen grains are released on the top of the surface of the water. They are carried by water currents and reach the female flowers. So, and the Vallisneria, you can see, so they are usually following this method. And the sea grasses, female flowers remain submerged in water. Pollen grains are long and ribbon-like. They are carried inside the water and reach the stigma. Okay, so that example, in exam it may ask name flowers which are doing pollination by water. Okay, so in MCQ format they can ask. So water, here the pollen grains, most of the water pollinated species have a mucilaginous covering to protect from wetting. So it is totally covered. Just will take example of coconut. So outside cover is very hard. That protect the inside area. Okay, that we know. So wind and water pollinate flowers are not very colorful and do not produce nectar. They don't have any nectar. So here also you can see how the process is carried out. Next is the animals. The pollination, the agent is animals. So they are called entomophilic. They are called entomophilic. The pollination by insects, it is called entomophilic, and particularly bees is more common. So in that bees, butterflies, flies, beetles, wasps, ants, moths, birds. In that sunbirds and hummingbirds, bats, primates, arboreal, land rodents, reptiles, many things. Even humans also, they are responsible for this. Okay, so features of the insect pollinated flowers. So they are large, colorful, fragrant, and rich in the nectar. And nectar and the pollen grains are the floral rewards for the pollination. So nectar, generally, if you know that honeybee, they are collecting the honey in the nectar, that you know, and the small flowers from the inflorescence to make them visible. So that is why flowers have their special appearance, that the fragrance they have, and that is why you can see the butterfly, it's sucking the juice from the flower, and they are collecting. Honeybee, just honey, we know that honey, mud, that we are aware. So when the animal comes in contact with the anthers and the stigma, its body gets pollen grains. And when it comes in the contact with the stigma, it results in the pollen must attract, must stick with each other, and then only the pollination process will carry out. Okay. So here some plants provide safe places as floral reward to lay eggs. In that Amorphophallus, here you can see Amorphophallus, that is, it has the tallest flower of 6 feet. Remember this. You can see this on Discovery Channel. One program is there. In that also you can see the highest and lowest. In that one program is coming, you can see that Amorphophallus, that is the tallest flower of 6 feet. And moth species and the plant yucca cannot complete their life cycle without each other. So this is some additional information for you. So many insects consume pollen or nectar without bringing about pollination. They are called pollen or nectar robbers. Generally they are doing robbery, chori. They are doing chori from this process. Okay, so they are nectar robbers. Now outbreeding devices. So in that pollination, hermaphrodite flowers can undergo self-pollination, that is autogamy, and the continuous self-pollination result in the inbreed preparation that we discussed first and second. To avoid self-pollination and encourage the cross-pollination, there are some devices in the plants. So that self-pollination, cross-pollination, the one by one will see. So avoid the synchronization and arrangement of anther and stigma at the different positions. So first is avoiding synchronization. Here the pollen is released before the stigma becomes repetitive, receptive, or stigma become receptive before the release of pollen. So in that special arrangement we need to take care to avoid the synchronization. The pollen is reached before the stigma. Okay, and there's arrangement of anther and stigma at the different positions. So positions, you know that here positions. If you change the position, this process will stop. This is one type of outbreeding device. Cell incompatibility. Most important, cell incompatibility. Now, what is cell incompatibility in the outbreeding device method? So here it is a genetic mechanism to prevent cell pollen from the same flower or other flowers of the same plant from fertilization by inhibiting pollen germination or pollen tube growth in the stem. So this process is the cell incompatibility. So that once again I am trying to reading. It is genetic mechanism to prevent cell pollen. Cell incompatibility, remember, it is a genetic mechanism to prevent cell pollen from the same flower or the other flowers of the same plant from the fertilization by inhibiting pollen germination or pollen tube growth in the stem. The production of the unisexual flowers, generally they are monoecious. In the male and female flowers are the present of the same plant, that is unisexual. So one by one you can see, that is papaya. Papaya, you know that. That is a unisexual flower. Pistil interaction, the pollen and pistil interaction. So here, if you know this, the diagram we have already drawn in our 10th standard, if you know that. So pollen tube, antipodal, podal nuclei, egg cell, century, okay, and the longitudinal section. So here the pollen grains germinate on the stigma. How it germinate? Then the pollen tubes growing through the style, it will transfer. So here the pollen pistil interaction. The pistil, it is a process in which the pistil recognize the competitive or incompatible pollen through the chemical components produced by them. And if the pollen is compatible or right type, the pistil accept it central cell. This is a central cell. So in exam they may ask like this. So here male gametes and this is a pollen nuclei. So in exam also they can ask, they can provide this diagram and they will tell you to answer the particular label part. So here A, entry of tube into a synergy. B, you can see the discharge of male gamete into the synergy and the movement of the sperms, one into the egg and the other into the central cell. So it is if the pollen is incompatible or the wrong type, the pistil reject the pollen by the preventing, means the process will not be carried out. The pollination process will stop, fertilization there is no chance at all if the wrong type will be there. Okay, that you can remember. So here in the plants with said pollen grains, a two-cell condition, a vegetative cell and a generative cell. Two type of cells we know, vegetative cells and generative cell. So you remember this, all things you need to remember. Here you can see pollen pistil interaction, in that a plant breeder can manipulate pollen pistil interaction even in the incompatible pollinations to get desired hybrids. So generally the process will be carried out by the filiform apparatus, the synergy through filiform apparatus. The filiform apparatus is present at the micropillar part of the synergy to guide the entry of the pollen tube. So you can see the filiform apparatus. Here you can see filiform apparatus. So their function is to prevent at the micropillar part of the synergy guides the entry of the pollen tube. Now artificial hybridization. Artificial is totally man-made. So it is a cross. It is a crop and a cross hybridization improvement program in which desired pollen grains are used for pollination. Means healthy part we can use for the pollination, and this is achieved by the following two techniques: emasculation and bagging. Two methods we are using for the artificial hybridization: emasculation and bagging. So you can see here. So first emasculation, the removal of anther from the bisexual flower bud or a female parent before the anther dehiscence. Okay, and bagging, here the emasculated flower are covered with a suitable bag. You can see here the bagging process and emasculation, the particular part is removed or anther is removed from the bisexual flower. So that process will be carried out. Unisexual flowers, there is no need for emasculation because it is only one, and the female flower buds are met before the flowers open. When the stigma becomes receptive, the pollination is carried out using the desired pollen and the flower is repacked. So double fertilization we need to understand. In exam it may ask double fertilization is very important. So by step we try to understand. So here different steps are given. Suppose the pollen grain is transferred to the stigma. After that the sperm will be entered. Then how the fertilization process will start. The ovules are already there. They fuse the ovum, the sperm nuclei, the synergy, that is microfill, eggs are there. The pollen nuclei will start embryonic sac development, and then it will start the fertilized embryo sac. So here degenerating synergies, the zygote formation will start. The PEC, that is and the PEN, you can say PEN, that is primary endosperm nucleus. PEN is a primary endosperm nucleus. You can see 3N, three numbers are there, and you can see PEC also. I will show you where it will be. So primary endosperm nucleus, PEN will develop. double fertilization. It is called double fertilization. Central cell after triple fusion becomes the primary endosperm cell (PEC) and develops into the endosperm zygote develop into embryo. Got it? So like this the process will be carried out. The post fertilization events. So in the endosperm development first, after that embryo development, ovule into seeds and ovary into fruit. This we know. The germination process we are aware. So in that endosperm development first, that is double fertilization. After that embryo development, then ovule convert into seeds. Ovule convert into seeds and ovary convert into fruit. Ovule convert into seed and ovary convert into fruit. So that you need to understand. So this is the step 1, 2, 3 and 4. So this is the endosperm development and embryo development. After that, the globular embryo and mature embryo, how the process will be carried out. Cotyledons, radicals. So in that plumule, ovule to seed and ovary to fruit. So that's we need to remember. Okay. So now endosperm development. So endosperm development is the primary endosperm cell divides the repeatedly to form a triploid endosperm tissue. You can see already we have seen. So ovule convert into seed. Okay, so it is a process of seed germination. And endosperm cells are filled with a secure food material, and they are used for the nutrition of the developing embryo. So then there we are getting the nourishment, we are getting the nutrition. So and that in common endosperm development, PEM, there is a primary endosperm nucleus undergoes successive nuclear divisions to give free nuclei, and the stage is called free nuclear endosperm, F and E. Free nuclear endosperm. Number of free nuclei vary greatly, and endosperm becomes cellular due to the cell wall formation. So here cell wall formation will be there. That is why plants have cell wall. Remember this. Okay. So and tender coconut water is a free nuclear endosperm made up of thousands of nuclei. Generally, the water we are drinking, if you know the coconut water, and surrounded water canal, it is a cellular endosperm. Now embryo development. Embryo develops at the micropillar end of the embryo sac. Generally, they are generating. The most zygotes develop divide only after the formation of some endosperm, and this gives nutrition to the developing embryo, and it is mostly in the monocots and dicots, and seeds differ greatly, but embryogeny, that is early embryonic development, is similar. So here zygote, pro-embryo, globular, heart cell and mature embryo. This is the step. Just you to understand zygote, pro-embryo, globular, heart cell and mature embryo. So this is the whole process of embryonic development. So here the embryo development, dicotyledons embryo, that is 2. In that plumule, that is stem tip, and the radicle, that is the root tip. Remember this epicotyl, that is the plumule, stem tip, and hypocotyl, that is the radicle, root tip, and the root tip is covered with a root cap. That generally we know this. And this process, monocotyledons only one cotyledon will be there, and in that it is situated lateral to the embryo axis, and at the lower end the embryo axis has the radicle and the root cap enclosed by the pseudo and undifferentiated silk. So here epicotyl, it has the shoot apex ovule. So in the non-albuminous seeds and albuminous seeds. So what is the difference between them? So mature seeds have two types. There is a non-albuminous seed. They are no residual endosperm, as it is completely ensured during embryonic development. For example, pea, groundnut and bees. And albuminous seed, they are retain some endosperm. And for example, wheat, maize, barley, custard, coconut and sunflower. So these are albuminous seeds and non-albuminous seeds. Okay, so like this, generally we know the embryo. This is the embryo, how the germination process will start, and seed coat, which is protecting them, and food store there for the nutrition. Now here one important thing we need to understand, that is dormancy. What is dormancy? As the seed matures, it becomes dry by reducing water content 10 to 15% moisture by mass. The metabolic activity of the embryo slows down, and it may enter a state of inactivity, that is called dormancy. Okay, that this generally the outer layer is become very hard, and under unfavorable or favorable conditions, the moisture, oxygen and suitable temperature, they are germinated. So for this type of seed, they need more time for the germination. So that is why, if you know that pea, gram, okay, wheat, so they need more time for the germination. Suddenly within few days it will not germinate. It will minimum require 6 to 7 days for the germination. So this type of examples you advantage of seeds. Different advantages are there. Seeds from the ovule. Here also the hard seed coat protect the embryo. How the seed coat is protecting the embryo. Then viability of the seeds after dispersal. So that also very much important. So here some information for you. If a few species, the seeds lost viability within a few months, seeds of many species live for several years. And some seeds can remain alive for hundreds of years. The oldest is that of lupine, that is Lupinus articulus, that is excavated from Arctic tundra. And the seed germinated and flower after estimated record of 10,000 years of dormancy. Oh, it's very long time. Our many generations will see this. Okay, so that is not possible, we will see. But it's mentioned in 10,000 years to take to germinate. That example you remember, lupine. Okay, so lupine takes 10,000 years to germinate for the dormancy. Fruit from the ovary. So ovary deposits the fruit, and pericarp wall of the fruit. So different two type of fruits are there: true fruits and false fruit. You will separate. What is this? Two fruits are there: true fruits and false fruit. True fruits means in this the fruit develop only from the ovary. It is a nature. If in this false, the thalamus also contribute to fruit formation. For example, apple, strawberry, and cashew. So they are false fruit. True fruits, we have example. Most plants are true fruits. And some species fruit develop without fertilization. Such fruits are called. Remember this. This is very much important for the MCQ and 1 mark question. In some species, the fruits develop without fertilization. Such fruits are called parthenocarpic fruits. Parthenocarpic fruits. For example, banana. In that, no fertilization. Without fertilization, the fruit will develop. So, parthenocarpic can be induced through the application of growth that mimics sexual reproduction. Here you can take some examples also. There is an apomixis, and the sectional part, the development of apomictic seeds. In such species, the diploid egg is formed without reproduction division and develops into the embryo without fertilization. So here many citrus and mango varieties are there. They are having a apomictic seeds. Generally, if you know this is a test tube culturing nursery. We know that. In that, the importance of the apomixis in the hybrid seed display nursery, if you know, in that the hybrid plant will develop. Okay, so thank you everyone. It is the end of this chapter, and most of the topics I have covered. You just go through your textbook and try to read chapter number 2. You will understand most of the topics we have covered. So chapter number 2, you will go through it. Important topics for the textbook. That is page number 20. A diagrammatic representation of LS of flower. It may ask. Then most of the structures are very crucial in the chapter because they may ask in 3 marks, even in 4 marks. Structures you need to draw, you need to practice for that, and this is also very much important. If you know this, already we discussed in this slide also, this part. Okay, that also you need to understand. So this is the end of the syllabus. Chapter number 2 is completed. If any problem would be there, we are here. You can come anytime. You can give us some feedback for this, and we'll try to solve your questions, your queries. So thank you. Have a nice day for this particular chapter, and we are conducting different exams also, so you will understand what type of paper